Hello, I'm Mr. Baker. This is the second section of my unit on evolution. We just got done talking about how different pressures, evolutionary pressures, or sometimes called selective pressures, change the different traits that are being picked by natural selection, right? Now we're going to talk about how those differences can actually arise, right? So how does variance occur? Right? And we're also going to look at how different types of movement into and out of populations can change how many of each allele is present in that population. All right, so let's get started with some old stuff. All right, remember that mutations are a easy way for differences in a DNA sequence to occur. They can happen via just errors in the DNA replication process that don't get fixed, or they could be caused by mutagens. Right? Mutagens can be chemical or radiation. Right? My obvious, super fun pop culture references are reading the wrong way. This guy's Spider-Man, right? Spider-Man is from a mutagen, a irradiated spider, so radiation mutagen, right? Or it could be chemical, like the Flash, who is exposed to different chemicals to give him his superpowers. Now, here in the real world, those aren't a thing, right? So we could look at UV radiation and sunburns causing mutations, or we could look at chemotherapy causing mutations and killing cells, specifically cancer cells, but it's still a mutagen. Remember that if it happens in a germline cell, it can affect the offspring, right? And that's the ones that we care about here in evolution because that's going to allow different amounts of each allele into the next generation. Remember our different types of mutations. Right? For my class, we already know all these. Maybe you don't in your class. Maybe you don't need to. That's all right. We've got silent point mutations that don't really change anything, but can change the DNA sequence. Right? So these aren't going to affect the allele frequencies because there's no change in the genotype or the phenotype. Right? But they could add up and end up causing a larger change. Most of the time, these have no impact, right? Then we've got missense and nonsense point mutations, which can change the amino acid being produced or remove an amino acid. They could also change the traits that are being actually evidenced into the phenotype, right? Because if you don't have an enzyme that does a specific thing, you might not get that trait, right? Then we've got insertion and deletion, the two frame shift mutations, where a new amino acid gets added to the chain or removed from the chain that is supposed to be made, and it makes a new amino acid, makes a new protein. That protein is going to do something else or may not function at all, right? It just depends on what actual mutation occurs. Then we've got inversion and translocation. Remember that translocation is what happens in crossing over, right? So it's one that normally happens. We'll say normally here, right? As in it happens all the time. And an inversion is just when a couple switch spots. So our first big concept here is genetic drift. What I want you to get out of it if you're in my class is that it is a change in allele frequencies due to chance. And it affects smaller populations more often. Because remember, like when we're taking averages, right? If there's lower amounts of numbers, a lower count of numbers, any change to those numbers has a bigger impact, right? So if we have, let's say, my green and brown frogs here, if those green frogs are able to camouflage into their surroundings and the brown ones aren't, that's going to be a directional selection, right? And then maybe those tan frogs are able to also camouflage into their surroundings. I know it's kind of hard to see because of YouTube's compression, but there's brown, there's tan, and there's a green frog here. So we've got two phenotypes, two morphs, if you will, that are surviving, and the green ones aren't, right? It's just due to chance that some of them are able to camouflage more. It's not due to any big changes here. It's not due to any weird mutations that are occurring. Just something happens, right? And the overall population isn't changing that much, but in the next generation, there's going to be less brown frogs because they don't survive to reproduce, right? If you can't reproduce, you're not passing on your traits. So due to chance, genetic drift is due to chance. Easy way to remember this is the last number in genetic is a C. The last letter, not number. The last letter in genetic is a C. Chance starts with a C. Right? Try to give you mnemonics when I can. The bottleneck effect is when something happens to the population as a whole. An earthquake, a tsunami, a meteor hitting the earth, right? A big thing occurs, right? And the allele frequencies change because only a small amount of that population survives. So this is always a loss in variation. There are less traits to be passed on, and so the new generation doesn't have as many traits as the old generation does. That's not to say that whole traits will get wiped out, but maybe specific alleles don't get passed on in the same frequency, right? So we've got our actual bottles here, right? You pour out a bottle with these marbles in them, and the surviving population has a different allele frequency than the original one. So you can look at that population on the side that is marked original, right? There's a lot of yellow marbles there and not a lot of red. But when the bottleneck event happens, when it gets poured out, more red ones end up in the surviving population. That's what we're going to call that because they survived the catastrophe. But only one yellow one survived. Who knows why that happened? It was a catastrophe, right? Insurance companies might call that an act of God, right? It's going to be something random that happens, but it's not due to chance. It's due to catastrophe. I know both start with a C. I'm sorry. Maybe I'll find a different word for that when I'm teaching it in my class, right? But that new surviving population, there's all those red marbles, right? So when those marbles reproduce and pass on their traits, we've got a whole lot of red ones in the new population where there's a much fewer number of yellow marbles. So loss of variation, always with bottleneck effect, it's always due to a catastrophe. This image is kind of how I think about it in my head to help me remember it. And then we've got founder's effect. Founder's effect is always due to migration, right? Specifically, immigration with an E. Immigration with an E is exit. Immigration with an I is into, all right? Those are two vocab words in my class that are going to be in this unit. They're good to know just in general. But there are a change in allele frequency due to migration. So you can see that the two populations here, right? We've got a bunch of bunnies with some gray and some brown fur. Some gray bunnies leave the population to start a new population. They cross this river. So the new population only has gray bunnies. And over time, those two bunny populations may change and be unable to reproduce with each other down the road. All right. For now, they're still the same species, but the population itself has lost variety because the new population only has gray bunnies. Right? Maybe they've got a recessive allele for brownness somewhere in there, but they all have that dominant allele for gray. So it's very unlikely that new brown bunnies will be made, right, through reproduction. Remember our heterozygous crosses that we had a couple units ago? 25% chance, but if they don't make heterozygotes in the next generation, maybe they make only homozygous dominants, then that allele frequency is going to get lower and lower and lower, right? With founder's effect, we also see a big population kind of becoming a small population, but remember, it kind of becomes two smaller populations because the existing population doesn't die, they just split. So genetic differences arise from mutation or from long-term changes in the gene pools when the population itself splits or changes, right? Immigration into a population will increase the genetic variance, whereas immigration with an E out of is going to lower the allele frequencies because there's going to be less of them, either through a bottleneck effect or through a founder's effect. It's going to lower it, right? Like I said, this is for my class specifically. Your class may not need to know all those, but I hope this helped.